From the quiet hum of Russian laboratories to the hopes of countless cancer patients, the journey of the EnteroMix Cancer Vaccine stands as a testament to innovation, perseverance, and the cautious optimism that defines modern medical breakthroughs. Developed on the heels of cutting-edge mRNA technology, EnteroMix is not just another experimental therapy. It represents a bold step in the global race to transform cancer treatment into a more precise, less punishing experience. But how did scientists take this vaccine from concept to preclinical promise? What challenges lie ahead before it can change lives around the world? Let's follow the path of EnteroMix, from its earliest days in research to the daunting threshold of clinical use, and explore the multifaceted challenges it faces along the way. The story of EnteroMix begins with a foundational shift in cancer immunotherapy. Unlike traditional vaccines, which are typically designed to prevent infectious diseases, therapeutic cancer vaccines aim to train the body's own immune system to recognize, target, and destroy cancer cells. EnteroMix leverages the power of personalized medicine, utilizing mRNA technology tailored to the unique genetic fingerprint of each patient's tumor. In practice, this means that RNA is extracted directly from a patient's tumor cells, decoded to identify key cancer-specific antigens, and then used to create a customized vaccine. The resulting mRNA vaccine, when administered, instructs the patient's immune system to mount a focused response against their own malignancy, ideally sparing healthy cells and reducing collateral damage compared to conventional treatments like chemotherapy and radiation. Before EnteroMix could be considered for use in humans, it underwent rigorous preclinical testing. These studies, conducted both in vitro and in animal models, sought to answer critical questions. Is the vaccine safe? Does it prompt a meaningful immune response? Can it shrink tumors without causing severe side effects? The results have been promising. EnteroMix demonstrated a remarkable ability to reduce tumor sizes by 60 to 80% in preclinical settings, while also improving survival rates in animal models. Importantly, these effects were achieved with minimal toxicity and a favorable safety profile, key criteria that paved the way for further development. Such outcomes fueled considerable excitement among researchers and clinicians who see in EnteroMix a potential to overcome some of the biggest hurdles in cancer therapy: specificity, efficacy, and tolerability. Yet, the leap from preclinical success to real-world impact is never simple. As of now, EnteroMix has completed its preclinical phase and is awaiting regulatory approval from the Russian Ministry of Health to enter clinical trials. The roadmap for clinical testing is both methodical and demanding. The first phase, Phase 1, will focus on assessing safety in a small cohort of volunteers, closely monitoring for adverse effects and immune reactions. Subsequent phases will widen the net. Phase 2 will determine optimal dosing and begin to evaluate the vaccine's ability to control cancer in a larger patient group, while Phase 3 will enroll thousands of participants to compare EnteroMix's effectiveness against standard treatments or placebos. Each phase is expected to span several years, reflecting the meticulous pace required to ensure patient safety and scientific rigor. Designing these trials involves a host of technical and ethical considerations. Will the studies be randomized and double-blind, minimizing bias and maximizing reliability? What endpoints will be used to judge success? tumor shrinkage, progression-free survival, overall survival, or quality of life. So far, the primary benchmarks include reduction in tumor size, extension of survival times, and the incidence and severity of side effects. Patient selection, informed consent, and continuous monitoring of adverse events will be strictly enforced, in line with international standards for clinical research. What sets EnteroMix apart technologically is its embrace of the personalized vaccine paradigm. Unlike one-size-fits-all therapies, each dose of EnteroMix is, in essence, a bespoke medicine. By analyzing the unique mutations and antigens present in an individual's tumor, scientists craft a vaccine custom-built to provoke a targeted immune assault. This approach holds special promise for notoriously hard-to-treat cancers such as colorectal carcinoma, glioblastoma, and melanoma, the very cancers EnteroMix is initially targeting. If successful, this model could redefine the landscape of oncology, shifting from generalized regimens to highly individualized protocols. However, the road ahead is not without obstacles. Despite compelling preclinical data, the breadth of testing is still narrow. Large-scale human trials have yet to be conducted, and the current data are limited to select cancer types. There remain unanswered questions about how EnteroMix will perform across diverse patient populations, various cancer subtypes, and in combination with other treatments. Furthermore, the costs and logistics of manufacturing individualized vaccines on a broad scale present significant challenges. Personalized medicine, while powerful, demands sophisticated infrastructure for rapid genetic analysis, mRNA synthesis, and timely delivery, a tall order even for well-resourced health systems. For patients and healthcare systems alike, the stakes are high. If EnteroMix delivers on its early promise, it could usher in a new era of cancer care, one where treatments are not only more effective, but also less toxic, reducing the physical and emotional toll on patients. The potential for lower overall treatment costs and improved quality of life is immense, especially when compared to the often debilitating side effects and high expenses associated with chemotherapy and radiation. For public health, a successful rollout could mean greater access to innovative therapies and a meaningful reduction in cancer-related mortality. Globally, EnteroMix enters a competitive and rapidly evolving field. Other mRNA-based cancer vaccines, notably those in development by Western pharmaceutical giants, are also advancing through clinical trials. Each platform brings unique strengths and faces similar hurdles, chief among them the need for robust, reproducible results in diverse populations and the challenge of scaling up production to meet demand. EnteroMix's success will be measured not only by its clinical outcomes, but by its ability to integrate into healthcare systems, remain affordable, and maintain supply chain integrity. Throughout this process, the legal, ethical, and communicative dimensions cannot be overlooked. The approval of any new medical intervention, particularly one as novel as EnteroMix, is subject to stringent regulatory oversight. Clinical protocols must adhere to ethical guidelines that protect patient autonomy, ensure informed consent, and provide for ongoing safety monitoring. Potential side effects and risks, while appearing minimal in preclinical studies, must be transparently reported and vigilantly tracked during human trials. In parallel, the medical community and media bear a responsibility to communicate developments accurately, avoiding excessive hype, and ensuring that patient expectations are grounded in evidence. As EnteroMix stands on the cusp of clinical testing, the collective hope is palpable but measured. The journey from laboratory breakthrough to bedside reality is long and complex, fraught with scientific, logistical, and ethical hurdles. Yet, with each carefully designed trial and every new piece of data, the vision of a more personalized, effective, and humane approach to cancer treatment comes closer to fruition. Whether EnteroMix will ultimately fulfill its promise remains to be seen, but its progress so far has already ignited fresh optimism in the enduring quest to conquer cancer.